glucose, exokinase is the enzyme, we have glucose, we have ATP. This kind of a direct reaction between glucose and ATP to form ADP and glucose 6-phosphate. We'll see this reaction again on Friday. Right? So that's a reaction where you don't really have an intermediate. There's no covalent intermediate. There's no intermediate in terms of the structures of the substrate. It's kind of just in and out attack and formation of products. Right? But if I show you a reaction like this one, where we have glutamate going to glutamine, right? Most biochemists would be hard-pressed to understand exactly how ATP is used in this. Right? It's not a situation where you could say, oh, I have glucose, I have ATP, I know it's glucose 6-phosphate, the nucleophile must be the oxygen on the CH2OH at position 6. It's going to attack this phosphorus and ATP, right? You can kind of get a picture of what it is. But if it's this reaction here, right, you know, good luck figuring out what it is, because there's no obvious thing that's going on. Somehow this, you know, ammonia comes in here and substitutes for that oxygen. I don't know how that happens. Right? Unless we look here and we see how it happens. Right? So ATP is converted to ADP and phosphate, but it's not a hydrolysis where phosphate is formed. ADP is formed and the phosphate ends up attached to the gamma position of the side chain of glutamate. Right? So we get glutamyl phosphate. And glutamyl phosphate is an activated compound. Right? It's got this high energy linkage, and then ammonia can attack that carbon, and the phosphate can form, and this is this favorable leaving group, all that resonance, and you get glutamine. So it's just this two-step reaction. So sometimes the authors of the book are nice to you, and me, and write this out, and sometimes they're not. Right? In which case, we don't really know what the mechanism is, and you know, we have enough experience in biochemistry to take a decent guess that it would be something like this. But we wouldn't know unless we did experiments to try to isolate an intermediate, really track the reaction and figure out what was going on. You should be able to do stuff like look at if glutamate reacts with ATP to form this compound, you should be able to look at the structures of these compounds and say, what is the nucleophile exactly? Like which atom is it? What's one of the oxygens on this right, the terminal gamma carboxylated glutamate? What is the electrophilic center? Right, the phosphorus, the beta gamma phosphorus of ATP. So that you can name what those things are. Same for here, what is the nucleophile and what is the electrophilic center? Yeah?
So in the first step of the reaction with ammonia, where does the third hydrogen end up on the phosphate in the end? Because the product has NH2.
Probably, yeah. Or it could get, depending on the pH and what else is around, it might end up on the enzyme, on an enzyme group. But if that happened, if we need to complete the cycle, it'd probably have to come back off the enzyme group. So yeah, so that's a you know you're pointing out an ambiguity around writing these reactions, which is that we don't always pay perfect attention to making sure that we really understand where where everything is coming and going from. And then you just kind of have to input some kind of common sense about enzyme reactions to think, well, it's going on a solvent, maybe it came from water, you know, maybe this PI got protonated, like you suggested. That's a perfectly good explanation. This is a bit of nomenclature, just because ATP is so important. You hit these reactions all the time in biochemistry, so you need to know the names of them. This is showing an attack of this is 18O is a radioisotope of oxygen, which would be used in experiments to track the products. It's not really important for this slide. But you can imagine an attack of an oxygen nucleophile linked to something on the gamma or the beta or the alpha phosphorus atoms of ATP. And here you're forming ADP and transferring the phosphoryl group, the PO3. This PO3 part would be transferred to here. Right? Here's PO3. This would be a phosphoryl transfer. Right? If you went here and you formed AMP instead, and you've got the whole pyrophosphate part, it would be a pyrophosphoryl transfer, obviously enough. And then the other common reaction is attack at the alpha position to release pyrophosphate and form this adenine ribose phosphate linked compound. Right? And here we basically transferred all of this. And so this is called adenylate transfer. Bit of a tongue twister there. So when we form pyrophosphate, not just in this reaction, but any reaction that forms it, there's this special bonus in cells, because almost all cells have this enzyme called pyrophosphatase, and that takes PPI and cleaves it and hydrolyzes it, binding water, binding pyrophosphate, forming 2 molecules of inorganic phosphate. So this PPI goes to 2PI by pyrophosphatase. This enzyme's almost always around. So if you form PPI in a reaction and then pyrophosphatase immediately removes it to PI, then by mass action, you can help drive the formation of more of this. Okay? So it's a way of immediately helping to drive a reaction if you form PPI. And this is very important in, for example, nucleotide synthesis. This is the synthesis, in this case, of RNA via template-driven reaction, where we're adding this G and pairing to a C on the other strand, for example, or whatever the other strand is. And so 3'OH attacks the alpha phosphorus of GTP in this case, right, and forms the phosphodiester linkage to lengthen the RNA chain, and it produces pyrophosphate, and then the second anhydride bond is broken here to generate 2 molecules of inorganic phosphate. Right. So this lowers the pyrophosphate concentration, so it's going to improve delta G because it's going to improve that Q term. Remember, from delta G equals delta G0 plus RT ln Q, and Q is the concentrations that are actually in the cell. If the PPI concentration is low because pyrophosphate eats up whatever PPI forms, then that's going to help the reaction drive in the direction of forming more pyrophosphate. Right? So this is a big driver for synthesis of nucleic acids themselves. Questions? Okay, let's move on. All right, so the last sort of topic in the sort of conjunction of the whole collection of things that we talked about here in the beginning of the class is something about redox reactions. It turns out that you know, for all the attention that we're going to give right in the beginning, we're going to give a lot of attention in glycolysis. Just starting with glucose, and we're going to track through this pathway, and we're going to, you know, we're going to learn all the structures of the compounds in the pathway, and see where the ATP is made on the enzyme, and think about which reactions are favorable and which ones are not, which ones are regulated and which ones are not. For all of that attention that we give to the glucose pathway itself, along the way, there will also be some redox reactions. And the redox reactions have the effect of moving electrons from fuel-rich molecules like glucose. And later in the term, we'll learn that it's the same thing for fats. These are fuel-rich molecules that are effectively burned in the cell. Right? And so electrons are removed from them. Okay? And so they're oxidized. Right? And they're transferred to some acceptor, which of course is then reduced, so you have a redox reaction. Right? So a lot of the redox reactions in cells have to be understood in terms of looking at the structure of a carbon substrate as you go through the glycolytic pathway, for example, and other pathways. So that we want to be able to recognize when we look at a carbon-containing molecule that's mostly uncharged, or if it is charged, it's not really relevant to this redox thing we're talking about, to see what is that an oxidized, how oxidized is that carbon-containing molecule, that sugar in the middle of the glycolytic pathway, as compared to some other carbon-containing compounds. And so this table is kind of a bit of a cheat sheet to tell you about that if you're not familiar. you know, there's no charges here. We're used to thinking of redox, perhaps, in terms of, you know, what you learned in freshman chemistry, iron 2, iron 3, oxidize it, remove an electron, go from iron 2 to iron 3. You know, your whole electron's been removed, so it's obvious what the redox state has changed. You've got a number on it that tells you it went from +2 to +3. So there's no numbers like that because there's no actual formal charge on these carbons. Nonetheless, we're saying that this carbon, this red carbon in ethane, is more reduced than this red carbon in ethanol. So that carbon got oxidized in the process of going from ethane to ethanol. And this gets associated with this number 7, which basically says if you've got a carbon bound to a hydrogen, we'll count both electrons as if they were part of the carbon. If the carbon is bound to another carbon, we'll count one of them, and the other carbon gets the other one. But if the carbon is bound to an oxygen, it doesn't get either one because the oxygen is more like electronegative. So this is, you know, this is pretty quick and dirty here. It's not really very accurate, but it provides a mnemonic or just a tool to say, well, you know, here there's only 5 little red dots around the carbon, and the number's lower, so lower numbers mean more oxidized. In this scheme of counting the electrons that are on the carbon, because they've basically been taken away. Right? So oxidation, removal of electrons. Okay? So this carbon is oxidized. Usually, when the carbon is oxidized, you can recognize if it's bonded to something electronegative, often oxygen, sometimes nitrogen. Right? So alcohols are more reduced than aldehydes and ketones, and acids are more oxidized yet. This is a little bit of a misnomer here, because usually we think of a carboxylic acid as being more oxidized than the ketone. So I think this table breaks down a bit there. Here you've got a carboxylic and acetic acid where the number is 1. Here you've got a formic acid and the number is 2. So this is only showing the limitations of this way of trying graphing things.